All right. Good morning, everyone. I'm Meenakshi. Welcome to our CCMMP seminar series. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are joining me from today. I pay my respects to their elders past, present, and emerging, and to the Aboriginal elders of other communities. A little bit of housekeeping rules before starting. It would be great if you could mute your microphone throughout the seminar, and there will be a question answer session at the end of the presentations. You can either unmute yourself and ask the question directly, or you can type it in the chat box. I can read it for you. And just to let you know, the seminar will be recorded, so it will be available on the CCMMP website. Today we have with us Professor Brett Collins. Welcome, Professor Collins from University of Queensland. A little bit about Professor Collins. After finishing up his training at Macquarie University in Sydney and a postdoctoral period at the Cambridge Institute for Medical Research, Professor Collins returned to Australia in 2006 to take up an NHMRC R.D. Wright Career Development Award at the University of Queensland. He currently holds an NHMRC investigator grant and is the director of the Center for Cell Biology of Chronic Diseases at the Institute for Molecular Bioscience at the University of Queensland. The career highlights of Professor Collins include determining the structure of the hetero-decameric AP2 complex that regulates receptor endocytosis at the plasma membrane, providing key insights into the Retromer transport assembly and the related commander complex. Over to you, Professor Collins. Thank you.
Great. Thank you very much, Meenakshi, for the introduction, and thanks to the Centre as well for the invitation to talk to you about some of our work. I'd just also like to acknowledge that I'm speaking here from Mianjin on the lands of the Yagara and Turrbal people as well. So anyway, given the quality of the speakers you've had in this series so far, it really is quite a privilege to be invited to give a talk to your centre. And I'm really happy to tell you about a protein complex that we've been working on for a few years, which is involved in a process called endosomal membrane trafficking. And this complex has a very grandiose name. It's called the commander complex, made up of quite a number of subunits. And yeah, I'm very excited to tell you about how we've been looking at this structure using a sort of broad range of different structural techniques. So saying that, if you do want to zone out and check your emails or anything while I'm talking, this slide almost gives a summary of what I'm going to be talking about. So it gives a bit of a spoiler. The commander complex is an essential protein complex. It's found in most eukaryotes, although it's not present in simple eukaryotes like yeast. And it's localized to an organelle called the endosome, and it's responsible for the trafficking of transmembrane receptors from the endosomal compartment. So it's a large complex made up of 16 different subunits. And some of those subunits are also mutated in rare developmental disorders, a rare developmental disorder called Ritscher Schinzel syndrome, or 3C syndrome, which refers to disorders of cranial, cerebral, and cardiovascular development. Okay, so I know that this is a cryoEM audience, and so just right from the top, I want to stress that I'm not a cryo-electron microscopist, at least not by training. I'm an X-ray crystallographer by training, but of course, as we all know, all structural biology labs these days are essentially incorporating cryo-electron microscopy into the structural biology work that they do. So I want to do two things today. I want to firstly tell you about this commander protein complex, what it does, and about how we've looked at the structure using different methods, so combinations of X-ray crystallography, cryo-electron microscopy, and also quite a lot of AlphaFold2 modeling. And then if I have time at the end, I'll just show a couple of examples of where this research is heading for us in the future. So this is actually, I think, the most important slide. I just want to point out that I haven't done any of the work that I'm going to present to you, at least not in the lab. And the work that I'm going to focus on today was really driven by several people, primarily by Michael Healy, a really amazing postdoctoral scientist in my team who began this work as a PhD student, Kerry McNally, Molly Chilton and Rebecca Butkovich, who are at the University of Bristol. And this has been an amazing collaboration with Pete Cullen's group, where these three postdocs are working. And we've also had an amazing collaboration with David Stroud's lab, who's now at Bio21, and his previous postdoc, Joanna Sachaz. But I won't have time really to talk much about the work that we did with them. So this is just some very basic textbook context, just to tell you about membrane trafficking. So we know that all eukaryotic cells are compartmentalized. So they have these membrane-bound compartments. And what this means is that all eukaryotes have evolved very sophisticated, very specialized protein machineries that control the sorting of lipids and transmembrane proteins between these different compartments. So lipids can often be transported between compartments by vesicular trafficking, or in fact by direct contacts through direct contact membrane contact sites. But with transmembrane proteins, they need to be transported between different compartments within membrane enclosed vesicles or membrane tubules. And so my lab is primarily interested in using structural biology methods to look at the types of protein machineries that control the sorting of these receptors and the formation of these vesicles and tubules. And we work mostly on proteins that are localized to this compartment called the endosome. And this is a major sorting station in the cell. So one of the main functions is that when proteins are taken up from the cell surface, they can either be sent to be degraded by maturation of the endosome and fusion with the lysosome, or different machineries that I'll talk about today can rescue these transmembrane proteins within vesicles or tubules to be recycled either back to the cell surface so that the receptors can be reused, or to other compartments like the Golgi complex. So this endosomal compartment is really important for maintaining the homeostasis of transmembrane receptors and also lipids within the intracellular transport system. So because of how important endosomal trafficking is for maintaining the homeostasis of receptors and organelles, it's also been implicated, or dysfunctions and mutations in these machineries have also been implicated in a number of different diseases. These include many diseases of immunity and infection, and in particular with bacterial and viral infection of cells. Much of the membrane trafficking machinery can also be hijacked by viruses and bacteria to infect the cell. It's involved in other diseases like cancer, but one of the most prominent areas where we see mutations or defects in membrane trafficking and endosomal trafficking in particular is in neurodegenerative or neurological disorders. And this includes in Alzheimer's disease and in Parkinson's disease, the most common neurodegenerative disorders. So the commander complex that I'm going to focus on today is very important for endosomal trafficking. And one of the cargo molecules for this complex is in fact the amyloid precursor protein involved in Alzheimer's disease. I won't talk much about that though, but more specifically, there are point mutations in the commander complex or subunits of the commander complex that cause this rare intellectual disability syndrome called Ritscher Schinzel syndrome, or also X-linked intellectual disability syndromes as well. Okay, so I just usually like to start my kind of structural talks with this really simplistic movie, just to kind of get across some of the key concepts of membrane trafficking for those of you that aren't in the field. So the sorting of proteins between one membrane and a target membrane is controlled by several types of protein machineries. And this involves, in particular, proteins that would act as cargo adapters. So these are soluble cytoplasmic proteins that bind to the cytoplasmic domains of the transmembrane receptors. And these usually bind to short peptide motif sequences within the cytoplasmic domains of the target receptors. And then there are other components. These can be an individual protein or larger protein complexes that act as like a scaffold that provide the mechanical capacity to deform the membrane, to form a vesicle or to form a tubule, and also to help promote the scission of that vesicle from the target or from the donor membrane. And this is such a simplistic slide, but of course, within this, there are also many other proteins involved, like snares involved in fusion, and the cytoskeleton, which is involved in targeting these vesicles from one organelle to another. But this is just giving the general concepts of vesicle formation. And in a past life, I did work on clathrin-coated vesicles, and these are undoubtedly the best studied of the types of transport vesicles. And this is a really nice animation just to sort of give you a pretty picture view of what this sort of vesicle formation can look like. In this case, you have a number of adapters that associate with the membrane surface. These then recruit transmembrane cargo proteins, and then they subsequently recruit this scaffolding protein called clathrin to make this clathrin-coated vesicle, which can be then severed off from the donor membrane and transported to the next compartment. In this case, clathrin-coated vesicles mostly transport material to the endosomal compartment. So to set the scene for the commander complex, I want to use Retromer as a quick comparison. So Retromer is something that our lab has worked on for quite a while, and Retromer and commander are actually two related but distinct complexes. These are both localized to the outer leaflet of the endosomal compartment. So Retromer here is shown as these little red squiggles, and it can recycle transmembrane proteins either from the endosome to the cell surface or the endosome to the Golgi complex. But it acts in a very different way to what I just showed you with the clathrin-coated vesicle. So instead of forming a spherical vesicle, the Retromer complex instead forms a membrane tubule. One of the biggest achievements from our lab in recent years was this cryo-electron tomography structure of the yeast Retromer complex forming membrane tubules. That was work done by Natalia Leneba and Alexei Kovtun, who are now over in Germany running their own group. Sorry. And I just wanted to point out here that the commander complex, like Retromer, is also localized to the endosomal complex, to the endosomal compartment. And I'll show you in a minute, it's distantly related. The sequences are very dissimilar, but it has some structural features which are highly related to the Retromer complex. But it transports very different types of transmembrane proteins, and we don't yet know whether it forms the same sorts of membrane tubules. The cargo molecules for the commander complex include things like the amyloid precursor protein, or members of the integrin family of adhesion proteins, and also lipoprotein receptors that take up high-density lipoproteins. And all of the cargoes that we know of for commander are recruited to the commander complex through sequences called NPXY motifs in their cytoplasmic domains. So back to just show Retromer, just to sort of give you an idea of how these tubular vesicular structures are formed. This Retromer complex, shown here in red, green, and orange, is made up of 3 proteins. So it's got VPS26 in green, VPS35 in orange, and VPS29 in red. This is the yeast complex, and in this membrane assembly, this Retromer complex forms these arch-like scaffolds that form these polymeric arrays that sit on top of this blue protein. And this blue protein is a protein from the sorting nexin protein family. And there are lots of different members of this so-called sorting nexin or SNCs protein family that work as adapters to recruit the Retromer complex. So here in this reconstituted complex, the VPS5 protein in blue actually forms a dimer through what's called a bar domain, which associates with the membrane surface. The Retromer complex sits on top, and this polymeric scaffold generates this pseudo-helical lattice that can force the membrane to form these tubular structures. And these tubular structures are the exiting structures that would carry the transmembrane cargo proteins away from the endosomal compartment. Okay, so Retromer is one of the key machineries, and it was discovered quite a while ago. It was discovered first about 25 years ago, and we now understand quite a lot about its structure and function. But when we started working on the commander complex, it was really only discovered maybe 5 or 7 years ago. And so far, or at least until last year, there wasn't much known at all about its structure. So as I mentioned, it's required for endosomes to cell surface trafficking of some quite important protein receptors like lipoprotein receptors and integrins. And it was actually partly discovered based on the fact that 3 of its subunits are structurally related to the Retromer complex. And if you look closely here, if you've got a good eye, you can see this red, orange, and green protein. These are related to that Retromer complex I just showed you. Okay, so now I'm going to go through sort of step by step and show you how did we come to that structure of this commander complex. And first up, I should just point out what commander is. So it's a 16 subunit assembly that altogether is about 600 kilodaltons in size. And as I mentioned, it contains 3 proteins, VPS35-like or L, VPS26C, which is related to VPS26 in Retromer, and VPS29. And this is actually shared between the Retriever complex here and the Retromer complex that I just described before. So the commander complex is made up of 2 subassemblies. The first subassembly is this trimer called Retriever, and the second subassembly is a subassembly that we just call the CCC complex, which is made up of 2 coiled-coil proteins, CCDC22 and CCDC93, and 10 members of this COMD protein family, which just stands for copper metabolism MER1 domain. So these 10 proteins are all different genes. They're encoded by different genes, but they're all about 200 amino acids long, and they contain this C-terminal so-called COM domain and N-terminal helical N-terminal domain. So together, these 10 COM proteins and these 2 CCDC proteins, and I'll show you also this 13th subunit, DEND10, form this CCC complex. And what we know about the different subunits of this complex has mostly come through proteomic experiments. This is one proteomic experiment that was done by David Stroud's group, by Joanna Sachars and Callum McConville and his group. So they tagged one of the subunits, COMD9, with a flag tag, isolated that, and performed mass spectrometry. And this is something that we see very consistently, no matter which subunit we tag. We always pull out the known 16 subunits as the primary hits in these sorts of proteomic experiments. So that immediately suggests that these 16 subunits are probably found in a fairly stable assembly with each other. And apart from these core subunits, there are other peripheral proteins that seem to transiently associate with these core proteins. One of the most important ones that I'll come back to at the end is something called sorting nexin 17. And this is one of these sorting nexin proteins which acts as an adapter. So in this case, it's an adapter protein that binds to those NPXY motifs in the cargo proteins and recruits those cargo proteins to the core commander complex to be transported. One of the other key protein complexes associated with this assembly is something called the WASH complex. And I know that there's lots of acronyms and names here to remember, but the WASH complex is involved in activating the ARP23 complex. And what that does is promotes the formation of branched actin filaments on the surface of the endosomal compartment that seem to be important for maintaining the recycling compartment domains of those endosomal structures. So now, just before I get into the structure, I just wanted to sort of point out what we do and don't know about the commander complex. So unlike Retromer, we don't know whether it makes membrane tubules. And for the moment, I'm still not really convinced that it does make membrane tubules like the Retromer complex. But it does have some similarities. So the Retromer complex binds cargoes through sorting necks and adapters. It then makes tubules that transport those receptors away from the endosome back to other compartments. The commander complex also has a sorting nexin adapter called sorting nexin 17, which binds these NPXY motifs in cargoes like the alpha-5 beta-1 integrin and so on. And it associates with these domains of the endosome that are rich in filamentous actin. Somehow the combination of these actin domains with concentrated cargoes in the commander complex allows for the sorting of these cargo receptors away from the endosome to other compartments. And whether that involves membrane tubules is still something that's under debate. Just to show you in terms of the cell biology that we know about these proteins, this is just a simple immunofluorescence experiment here where we've labeled GFP tagged adapter protein SNCC17, VPS35L in red, and the Retromer subunit VPS35. And you can see where you get these white dots. All three proteins are strongly co-localized in these small endosomal domains of the cell. Similarly, we can tag GFP SNCC17 with VPS35 and FAM21, which is a subunit of that actin modeling complex called WASH. And these three proteins are all also co-localized within these same membrane domains of the endosomal surface. So the summary is there's an endosomal domain enriched in actin where the commander complex engages these receptors for endosomal recycling. Okay, onto the structure. So I'm going to start with these 10 proteins, the COMD proteins. So as I mentioned, there's actually 10 distinct genes. These are homologs. The sequences are not very similar, so they have diverse sequences, but they do share the same kinds of domain structure with this C-terminal COM domain and N-terminal helical domain. So a number of years ago, Michael actually solved a crystal structure of one of these proteins, COMD9, and this was published in eLife a while ago, and this was a collaboration with Sean Lott's group at the University of Otago. And what you can see here is that these proteins were immediately clear to us that these proteins must form obligate dimers. So the C-terminal COM domain sort of makes this hand-shaped type structure of beta strands and alpha helices, whereas the helical N-terminal domains are positioned off to the side of this central dimerizing domain. But even when we solved this structure, we knew actually from our own biochemistry work that this was probably actually an artifact. And in fact, what this was showing is that the COMD proteins have to form dimers, but the reality is they actually form a series of heterodimers and not homodimers, which is what we found when we just expressed a single protein in E. coli. So I'm going to skip a lot of years of biochemistry and hard work that Michael Healy did, where he really dissected out very nicely what were the different combinations of the potential COMD proteins, and just jump straight to a crystal structure that he was able to solve. So from all of the biochemistry he did, he could show that there were very specific arrangements of these 10 COMD proteins, and that they would always assemble in very set stoichiometries of set subcomplexes. And one of these set subcomplexes was made up of COMD7, which forms a dimer with COMD9, and COMD5, which forms a dimer with COMD10. And together, these two dimers, made up by that beta sandwich sort of handshake, then form a beta extension to make a tetrameric structure of the 4 proteins. And I'm not going to go into all the molecular details here, but essentially the sequences in these proteins that are found at the interfaces between different subunits dictate a kind of common principle for the organization, but also a specific pairing between the different subunits. And when you extend that to all of the 10 proteins, as I'll show you in a minute, you end up with a hetero-decameric structure made up of all 10 of these proteins assembled into a ring. So this is where I wanted to introduce AlphaFold. So we solved this crystal structure in 2019. That's quite a while ago now. And just after we solved this structure, AlphaFold came out. And obviously, all of you will be sort of familiar with the hype of AlphaFold when it first came out. It was solving protein folding. It was the solution to the 50-year-old grand challenge in biology. Became the method of the year in 2021. So there was a lot of hype about AlphaFold. I think clearly we now know it certainly didn't solve the protein folding problem, at least not from a physical perspective. But from our experience, it certainly has become a very powerful tool in structural biology. Of course, after all of this hype, there's obviously been a lot of backlash as well. So AlphaFold isn't going to revolutionize drug discovery, according to some people, and we should be questioning the hype about AlphaFold. And there's some truth to all of this, I think. But at least from a personal perspective, I'm going to say outright, I've actually personally drunk fairly deeply of the AlphaFold Kool-Aid. I really do believe that this is a revolution, or these types of methods anyway, a revolution in structural biology that are at least on a par with the new methods of cryo-electron microscopy and even with the original invention of X-ray crystallography. But I would also stress I haven't drunk quite so deeply that I don't think it is perfect. It certainly has a lot of flaws and a lot of limitations. And I think as long as we're aware of what those limitations are, though, it can be an extremely powerful tool for us as structural biologists. So as I mentioned, we solved that crystal structure, and almost a month later, AlphaFold was released. And one of the first things that Michael did was just to put those 4 sequences into AlphaFold and see what happened. And I must admit, at this stage, I was a skeptic. But when we saw this prediction, it was exactly the same as our crystal structure. And it put all 4 proteins in exactly the same order as what we see in the crystal structure, and just matched it perfectly. I have to admit, I was pretty gobsmacked by how well AlphaFold worked with this prediction. And I would like to point out that these structures weren't in the training set that was used to train AlphaFold as well. So that was really a pretty amazing eye-opener for us. And I guess that sort of set us on the path to look at what else we could do in modeling the rest of the complex. So from this step, we could then move on to looking at the rest of the proteins. Actually, just before that, I would just like to give one quick shout out to Sergei Ovchinnikov's lab and Martin Steinegger's lab, who have really done an amazing job in democratizing the use of AlphaFold with the CoLabFold stream, which is something that we use all the time. Okay, so at this point, Michael and I just kind of went a bit crazy and modeling different things with AlphaFold to see what we could find. And one of the first things we found is that we could actually model a structure with all 10 COMD proteins and N-terminal fragments of the 2 coiled-coil domain proteins, CCDC22 and CCDC93. And this prediction puts together all 10 COMD proteins into this beautiful ring structure, and also positions the 2 coiled-coil N-termini that into structures that sort of wind their way through the rest of the COMD proteins. It's really quite amazing the way that this works. Here is one of what's called the Calponen homology domain of CCDC93, and that's the Calponen homology domain of CCDC22. These are regions that on their own are predicted to be unstructured, and then they would trail off to make the coiled-coil parts of these 2 proteins. And this prediction immediately kind of answered to us why, when we expressed these proteins in bacteria, we never got a full 10-mer structure. And it looks like it's because this decameric structure is actually held together by these two coiled-coil proteins that we weren't expressing in bacteria. But this is where Pete's lab, Pete Cullen's group, had more success. So they were able to then make a complex that had all 10 proteins with the two coiled-coil domain proteins in baculovirus and insect cells. So they were able to obtain some really nice homogeneous complex, get some very nice particles, and get some very nice 2D class averages. This data was collected on a Krios microscope at the LMB. One thing you will notice here is that although it has all of the coiled-coil regions of the CCDC proteins, you can't see these in the 2D class averages. And that just suggests that what we're really picking up in these particle picking pipelines here is the central COMD ring without the coiled-coil regions, or with the coiled-coil regions being very flexible. So this is the 3.2 angstrom resolution cryo-EM structure of the 10 COMD proteins with the CCDC22 and 29 and 39, CCDC22 and CCDC93 N-termini. And you can see it makes this ring structure where the center of this ring is made by the beta strands of the COM domain, with the helical N-terminal domain sitting on either side of that central ring. This decameric structure is made up of a series of dimers of particular COMD pairs, and they always arrange in exactly the same order around the structure. And the two CCDC calponent homology domains sit to the periphery of this structure, and they wind through the structure. It's not quite clear from this moving model, in exactly the same way that is predicted by AlphaFold. So the beauty of this cryo-EM structure in itself, I think, is amazing. But one of the also really amazing things is when you overlay this with the AlphaFold prediction of all of these 12 proteins, there's almost no difference. So AlphaFold has somehow managed to predict this very complicated hetero-decameric structure ab initio without any experimental data, and predicted it almost perfectly. In fact, to build this cryo-electron microscopy structure, we just took the AlphaFold prediction, docked it into the maps, and wiggled some of the domains a little bit, but there really wasn't a lot of rebuilding that we needed to do. So this is now taking us through the bit of this structure. So the 10 COMD proteins and the N-termini of these two coiled-coil proteins. If we, based on the success we had, we were now sort of more confident to move ahead and look at other bits of this. And first we looked at this DEN-D10 protein. And this is where AlphaFold really became quite powerful for us once again. Although we weren't able to get a crystal structure of DEN-D10 bound to the complex, or a cryo-EM structure, we could actually use AlphaFold firstly to scan through all of the other subunits to see where it was binding, and then use AlphaFold to get a very highly confident prediction of how it was interacting with this particular dimeric coiled-coil region of the two CCDC22 and CCDC93 proteins. And if you look here in blue is highly conserved parts of DEN-D10, and in the AlphaFold prediction, these extremely conserved regions are binding to these two coiled-coil fragments of the two CCDC proteins. And I just wanted to stop here just quickly and just mention, in terms of looking at AlphaFold predictions, what are the sorts of things that we've found are good ways of judging the quality of those predictions? When you do these sorts of multimeric predictions, the first thing we look at is something called the IPTM, or interfacial PTM score. And that gives you a sort of a number that's an average of the confidence of the predictions of the residues that are near the interface between the different proteins. But I think to us, what we found is almost the most important thing to look at are these predicted alignment error plots. And here shown in blue is one of these sorts of plots for this complex. And this is giving you a measure of the confidence in the structural precision of a particular amino acid if you align the structures on another amino acid. So if that score is very low, it suggests that those two residues are essentially rigidly located relative to each other in space. And so if you get good off-diagonal PAE plots that suggest different proteins are forming close contacts with other proteins, for us, that's usually the best predictor of whether you have identified a real complex. The other thing we do is obviously just overlay multiple models of predictions. And I think still, as a structural biologist anyway, I think there's no substitution for just looking at these structures in Pymol or Chimera or Coot, just looking at them and seeing whether they make chemical sense. If you align multiple structures, do they all look the same? These sorts of empirical or subjective judgments of the model, I think, are equally important. We also often compare structures across different species, but most importantly, I think no matter how good these predictions are, it's always essential to try and combine these with experimental structure validation or binding and mutagenesis. So, for example, in this case, we couldn't get a structure, but we could reconstitute these three proteins into a really nice complex, and we could use methods like ITC to show that they bound to each other with high affinity. And in cells, and I'm not going to show the data, we can mutate the interfaces between these proteins and abolish those interactions based on these predictions. So from there, we then moved on and we had a look at the predictions of the full-length coiled-coil domain proteins with DEND10. And you can see you have the N-terminal regions that extend into these hetero-decameric coiled coils with coil-coil bit 1, 2, then another V-shaped coil-coil region of coil-coil 3 and 4. One of the interesting things we often find in the predictions is the Calponen homology domain of CCDC22, sorry, those are mislabeled, it should be 22, is bound back into this groove between these C-terminal regions, and I'll come back to this a bit later. These parts of the CCDC proteins are where the COMD ring sits. So these parts are actually now threading through the COMD rings. So from there, the next bit we looked at was the trimeric retriever subcomplex. Unfortunately, we couldn't get a high resolution structure, but we were able to collect some low resolution cryoEM data that was unfortunately quite limited due to particle orientations, but we could get some low resolution maps and compared this to an AlphaFold model prediction of these three proteins, which matched very nicely. So in this complex, you have VPS35L in this alpha helical solenoid with VPS29 sitting at one end and VPS26C sitting at the other end. This is the N terminus of 35 and the C terminus of 35. So this is the part of the complex called retriever, which actually is structurally similar to the Retromer complex. But if you compare this cryoEM structure of the Retromer complex to this prediction and low resolution cryoEM structure, you can see that they look quite different, although they share the same morphologies or topologies in the different protein subunits. In particular, the VPS35L subunit is much more compact and compressed than the VPS35 subunit. Now, as I mentioned, VPS29 is a core subunit that's found in both of these different protein machineries. So apart from the difference in their overall shape, one of the really key differences in these two complexes between retriever and retromer is that it's not really shown very clearly here, but there's an N-terminal disordered loop in VPS35L, which comes back this way and crosses over VPS29 and makes a very intimate interface with the VPS29 subunit. And that's not seen in the Retromer complex. In fact, in the Retromer complex, where this sequence is associated is where Retromer interacts with various accessory proteins. So it's very clear from this that Retriever is actually using an intramolecular contact to block this site so that VPS29 can't engage those Retromer-specific accessory proteins. So here is this peptide sequence. It has what's called a proline-leucine motif binding to a very conserved pocket in VPS29. And initially, this was based on an AlphaFold prediction, also combined with this low-resolution cryo-EM map. But we then obtained a crystal structure of this peptide complex to 1.3 angstrom resolution. And you can see just by overlaying these two structures that the AlphaFold prediction and the X-ray crystal structure match each other perfectly. Honestly, as a crystallographer, if you gave me both structures, I wouldn't have been able to tell the difference or which one was the crystal structure. The last bit of this, before we put it all together, was how do the CCDC proteins, or how does this CCC complex associate with Retriever? And we found that this is occurring through this C-terminal region of the CCDC proteins binding to the Retriever complex. And I won't have time to go into detail here, but we've mutated all of these interfaces and shown that this is exactly how the Retriever interfaces with the coiled-coil domain proteins. So to get to this final structure, essentially what we then did was piece together cryo-EM structures of the COMD ring, cryo-EM structures of the Retriever complex, AlphaFold predictions of the coiled-coil domain proteins, crystal structures of VPS29, crystal structures of some of the COM domain proteins. We assembled these into an overall structure of the sixteenth subunit assembly. And when you put that all together, you end up with this model where the ring of COMD proteins is engaged by these central regions of the CCDC proteins and sits in this flexible extension between this kind of V-shaped part of the coiled-coil proteins. This then has a platform which is able to then bind down here to the Retriever complex. So basically what we have now is the overall structure based on experimental and prediction methods. And just to highlight here, despite the fact that some of these are predictions, we've mutated essentially every interface in this entire complex and tested those in vitro and in cell biology assays to really validate all of these different experimental and predicted interactions between the proteins. So this provides us with basically an overall structural model of the commander complex. But one thing I'll show you in a few slides is that we speculate that this may actually just be one static representation of a potentially quite flexible assembly. So we've got this structure, which tells us a lot about, obviously, how this assembly is put together. But one other thing we can do with that is go back to the known point mutations. Don't have to read all of this slide, but essentially there are mutations in both CCDC22 and VPS35L, as well as the WASH actin remodeling complex that lead to this Ritscher Schinzel 3C syndrome. And patients with...
Unfortunately, there's nothing from the screen. Oh, yeah, we'll have to give it another try when they send more protein. Yeah, it's disappointing, but it's the start. Yeah.
Sorry, was that talking to me?
No, I guess by mistake. Someone has...
No.
Sorry.
No, that's fine. Yeah, so we have all of these missense and deletion mutations in the CCDC22 and 35L subunits. One of the things that these patients also have is hypercholesterolemia. And we think, or at least the authors of that paper think, that that's due to defects in lipoprotein receptor trafficking, things like the low-density lipoprotein receptor. we can take our structure and take all of those known missense mutations and map them back onto the structure. And while there are some interesting ones that sort of sit in interesting regions, the majority of those mutations lie at the interface between the retriever subcomplex and the CCC subcomplex. And Koji Kato's group did some really nice proteomics where they took some of these disease-causing mutations. And I'm not showing all the data here, but just a couple of examples. But essentially what we find is that the mutations fall into two distinct categories. They either completely disrupt the complex. So for example, this alanine to threonine mutation in the VPS35L subunit essentially causes the whole complex to dissociate and probably to degrade in the cell. In contrast, this deletion mutant in VPS35L, which sits near the C-terminal VPS29 binding site, seems to keep the rest of the interactions intact. The rest of the CCDC and COMD proteins are all bound still, but VPS29 is selectively lost from this complex. So we either have mutations that cause complete disruption and degradation of the complex, or we have mutations that cause selective loss of particular subunits. And it's yet to be sort of studied in detail as to whether these different types of mutations lead to different kinds of disease severities. Okay, so don't have too much time left. I've got a few more slides to get through. I was mentioning just before I started this seminar that as we submitted this work early last year, we put the paper onto bioRxiv while it was under review. While the paper was under review, two more papers came out on bioRxiv from two other groups with similar structures to us. So as I mentioned in this title, the competition really did start to heat up early last year. So that was slightly terrifying, but also these two papers are now published back to back in Nature Structural Biology after our paper in Cell. And it's now really quite nice to see that all three papers are essentially validating and confirming the other work, so that all three structures are essentially showing the same sorts of things with some differences that I'll point out. This was quite interesting. So one of the groups here was led by Ezra Berstein and Zhe Chen at UTS Southwestern Medical Center. So we knew that they were kind of working in this space, but this group here from Finland, led by Markku Vajasolo, was a group that we had no idea were interested in this complex. So it was kind of a left field paper. But it is nice now to see both of these papers out, even though it did give us a few heart palpitations at the time. I don't have time to go into all of the differences, but one key difference that is seen is between our structure and the structure from Ezra Berstein's group and the group from Finland. So in this particular case, they've used cryoEM to solve the structure of the whole 6-stage subunit assembly. But to get this structure, they had to use cross-linking between lysine residues. And in fact, what they see is a much more compact, compressed structure compared to our structure or to the other similarly predicted structure by Ezra Berstein's group. And it does look like this compressed structure is really only held together by a single cross-link between CCDC93 and the COMD7 subunit, bringing that ring together closer to the coiled-coil proteins. So this really is the biggest difference between the three different structures. And this is an important caveat, but I'm not averse to the idea of using cross-linking, but I think we do need to be a little bit cautious in interpreting this compressed structure as the structure of Commander, because I believe that this is actually an extremely flexible protein. Interestingly, in the Finnish group's paper, they did collect data on the native complex without cross-linking. They weren't able to resolve a structure, but you can see in the 2D classes, you can see these ring domains of the COMD proteins with some very vague, very light density from the rest of the protein. suggesting that there's a lot of flexibility between the orientation of that COMD ring and the rest of the coiled-coil retriever part of the assembly. And this is something actually that we did speculate on in our paper. This is one of the supplemental figures, but we had a little arrow here suggesting that this COMD ring is actually sitting here tethered by two flexible linkers at one end and another flexible linker at the other, where this component homology domain of CCDC22 binds back to the complex. And this is just to show you what I think may be happening, and it's completely speculative, but I believe that there may even be a regulatory molecule, a regulatory interaction here that's able to release this part of the complex and allow for a large degree of flexibility in this structure. Okay, so I am running out of time. I might skip through these very quickly, but I just had a couple of slides with two examples of where I think some of this work is taking us into the future. And these examples are really highlighting how we can incorporate AlphaFold-derived predictions of protein-protein interactions with validation of protein complexes. One thing about this coiled-coil proteins, as I've mentioned, is they have these component homology domains, and these are actually known to interact with the actin cytoskeleton in other proteins. This is a cryoEM structure of filament bound to the actin cytoskeleton. And so when we use AlphaFold, we can actually predict a very nice complex between the CCDC93 CH domain and an actin monomer. And in fact, this CH domain of CCDC93 is the most highly conserved surface on the entire commander complex. And here, this is just an extremely speculative idea of if we had some sort of interaction like this, what would the rest of the commander complex look like if it was associated with the actin cytoskeleton? So that's something we think may be happening, and we actually now have evidence that it definitely does bind to actin. So we've designed point mutations based on this AlphaFold prediction, and Ritscher Schinzel has shown that with these 2 point mutations, she completely loses actin association between the commander complex and actin in cells. And then the last example is looking at how the sorting nexin 17 adapter can associate with the commander complex. And here again, using a whole series of AlphaFold predictions, Michael and I discovered that a C-terminal peptide sequence in SNCS17, shown in blue here, can bind to a pocket between the VPS26C and VPS35L subunits of the retriever complex. And if you want to know more about this, we've actually now published this work on bioRxiv, so you can go and read the bioRxiv paper. But we've also done a series of mutations and other biochemistry validation and cell biology validation to show that this is indeed exactly how SNCS17 is associated with the retriever complex. So here, for example, if we mutate an arginine in VPS35L that sits at this interface, we completely lose the binding of the SNCS17 protein. So that brings me to the end. So to conclude, I guess we have really now a solid understanding of the core assembly of this commander complex. And I just point out that, sorry about that. That AlphaFold really has been amazingly accurate in predicting a lot of these interactions, but we've also been able to correlate this with a lot of experimental and cellular validation. And we've been able to map the domains or the regions of these proteins that are altered by mutations that cause these developmental disorders. And in the future, as I've just alluded to, we're really interested in how commander can associate with the membrane, with cargo adapters, with the actin-related wash complex, and as I just mentioned, directly interact with the actin cytoskeleton. The DEND10 protein is interesting because it looks like it's bound and inhibited in what we call an inhibited conformation. So we really don't understand what this DEND peripheral subunit is doing, which we'd like to figure out. And we're obviously very interested in whether the commanded complex can undergo any other major conformational changes potentially triggered by releasing of these coil-coil domain proteins. And I'm going to finish there. As I mentioned, the work that I showed today was primarily done by Michael Healy. It's been an amazing collaboration with Pete Cullen's group, in particular, Kerry, Molly, and Rebecca. Shrestha did some of the work that I showed just towards the end. It's been a great collaboration with David Stroud and his postdoc, Joanna Sachars. Tristan was really useful, or really helpful at the start with doing some modelling with his older, and also funding from both the NHMRC and the ARC over the years. Thanks, and I'm happy to take questions.
Okay, Fred, beautiful talk. So do you have any questions? You can either unmute yourself and ask or write it in the chat box.
Sorry, I ran a bit long.
It's okay. Well,
Hello, Brett.
Jason?
I'm Jason from MIPS, and I have a question about the AlphaFold predicted interface. And you mentioned about you use this prediction to design some mutation of the construct to lose the binding affinity between actin and your complex. I'm interested if you put the mutated construct into the AlphaFold prediction, the outcome will be some nonsense result, or it's still kind of maintaining the basically
We haven't done it specifically in that case, but we've done it for a number of other things. And like you suggest, it just doesn't work. So unfortunately, well, this is the way that AlphaFold works. It's based on evolutionary or co-evolution of amino acids between conserved parts of proteins. And if you just make a mutation in the sequence based on what you think is interesting, AlphaFold just doesn't really care. If it's got enough residues around that are conserved, it will still build the structure and it will put that weird residue where it shouldn't be, but it will still kind of put it together because it's not really based on any sort of physical concept of, you know, you shouldn't have a negative charge with a negative charge or anything like that. It doesn't really care. So looking at point mutations at the moment just doesn't work. But I do think we haven't done this, but I've seen a couple of other labs do this, like combining these sorts of predictions or also experimental structures. Things like MD, for example, can maybe give you some clues as to whether if you mutate a particular amino acid, do you block it?
Yeah, thank you. It's really nice talk. I have a tiny biochemistry question. I think you co-express 10 common protein in the same cell system. How do you kind of like balance the ratio between each species, or it's all in one construct?
Yeah, that's a good question. So we've done it in bacteria. And so when we express all 10 proteins in bacteria, we always can purify them in these separate subcomplexes. And one of those is the one that we crystallized. And that's because we think we don't have those coiled-coil proteins to hold it together. But when Pete's lab expressed the 10 proteins plus the 2 coiled-coil proteins in insect cells, they can get that 12 subunit complex out quite nicely. Now, in terms of the stoichiometry, we know that the stoichiometry of each of the subunits is 1 to 1 to 1. But in terms of controlling the expression, there's really no way of doing that. They're all encoded into the same plasmids. So they should have all the same promoters, but it doesn't mean that some of them may not be expressed slightly differently. It's just that when you purify the complex, you probably wash away the folded proteins or non-associated proteins.
Thank you. Really nice talk, and exciting to see the AlphaFold roles in the structural biology. Really good. Thank you.
We have another question from Sarah. Yes, hello. Sarah has asked, do you think the true structure of the two complex published, the more open one and the compacted one, is somewhere in between? Have you done endosimulation to be able to help with identifying the dynamics?
No, that's a good question. We haven't done any MD simulations. I think in some ways neither our structure or their structure is the correct structure. I just think that the position of that COMD ring relative to that sort of V-shaped coiled-coil protein and the retriever complex is essentially randomly distributed around these very flexible linkers. So there kind of is no set structure. But it could be, as Sarah suggests, it could be that it is a little bit more compressed. This is going to sound a little bit, I don't know, arrogant potentially, but when we did the AlphaFold modeling, we didn't see any evidence at all of any, and we looked really hard, but we didn't see any evidence for any of those COMD proteins making any kind of contact with those coiled-coil regions or with retriever. And so it's not evidence necessarily, but certainly we saw no indication at all that there is a compact conformation that's dictated by any interfaces between those proteins. So I do think that that cross-link between two of the proteins is kind of an artificial cross-link. But yeah, depends on whether you believe more of the mass spec cross-linking or believe more the lack of any evidence in AlphaFold modeling.
All right, so that's all. Beautiful talk, Professor Collins. I really enjoyed.
Thanks, Meenakshi.
Yeah, that's all right. Thank you so much for attending today's seminar, and thank you so much for giving this awesome talk. Thank you.
Thank you. I'll
All right,
see you
see
later.
ya. Yeah, bye.